Hello, and thank you for joining us. The title of our presentation is Effects of Antibiotic Use on the Gut Microbiome and Links to Depression. What if the secret to better health, brain function, and longevity lies in the tiny organisms living inside of your gut? Well, emerging research suggests that the gut microbiome plays a crucial role not only in digestion but in mood regulation, immune function, and even in aging. The gut-brain axis is a bidirectional communication network between the gut microbiome and the brain. Changes in the gut microbiota have been linked to autism, anxiety, depression, and even neurodegenerative diseases. Today, we will present 3 research studies focusing on antibiotic use, its effect on the gut microbiome, and links to depression. We will give a brief background on the gut microbiome, antibiotic use, and depression. We will present the research studies, their methods and findings, and then we'll finish the presentation with our conclusions, applications, and future research.
The human gut microbiome, a diverse ecosystem of bacteria, fungi, and other microbes, does play a crucial role in digestion, immune function, and metabolism, and even in brain health. It is established at birth and influenced by diet. It helps to synthesize essential vitamins such as vitamin B and K. It breaks down foods, producing byproducts such as short-chain fatty acids, and maintains glucose and lipid balance. The gut-brain axis links gut health to brain function, possibly affecting neurological disorders. With this dysbiosis, it can lead to permeability issues that allows harmful signals to reach the brain, thereby contributing to conditions like anxiety, depression, and Alzheimer's. Antibiotics have transformed modern medicine by effectively treating a wide range of bacterial infections. They are essential in treating conditions that can otherwise lead to serious health outcomes, hospitalizations, or death. However, their overuse and misuse have resulted in a global rise in antibiotic-resistant bacteria. Misuse of antibiotics include incomplete courses, self-medication, incorrect prescriptions, thereby driving antibiotic resistance in an individual. Antibiotics can disrupt gut bacteria, affecting both the harmful and beneficial microbes in that system. Solutions include optimizing antibiotic use, developing new drugs, and even exploring alternatives like probiotics and vaccines. Emergent studies have investigated potential links between antibiotic use and mental health, such as depression and anxiety. Depression is a common mental health disorder affecting over 280 million people globally. And unfortunately, the COVID-19 pandemic has led to a 25% increase in global anxiety and depression rate. Depression is diagnosed by a comprehensive evaluation of symptoms, medical, and family history. It is characterized by persistent feelings of sadness, emptiness, or hopelessness. If left untreated, it can severely impact relationships, productivity, and increase suicide risk.
For methods, we focus on original research studies that focused on the disruption of the human microbiome by antibiotics and effects on depression. Studies were eligible if they included a control trial. The studies had a validated instrument to describe major depressive disorder or microbiome disruption. And we looked for articles between 2019 and 24. We ended up finding 7 original research studies and used the databases PubMed, Google Scholar, Aurora Library, and Cochrane Library. And here are the keywords that we used as well.
Rajabzadeh et al. Conducted a gut microbiome-wide association study to explore the link between gut microbial composition and depressive symptoms. This cross-sectional longitudinal study included 1,054 adult participants, aged 45 to 88, selected from the Rotterdam study, a large ongoing population-based cohort study in the Netherlands. Researchers collected fecal samples from the participants to analyze their gut microbiome composition and identify microbial taxa associated with depression. They performed 16S rRNA gene sequencing to identify bacterial taxa present in the gut. To measure depressive symptoms, they used the Center for Epidemiological Studies scale, CES-D. This study also accounted for various clinical and demographic variables such as age, sex, BMI, smoking status, and medication use to adjust for potential confounding factors in their analysis. The study found significant associations between certain bacterial species and depressive symptoms. If we refer to the graph on the right, it represents the association between these specific microbial taxa and depressive symptoms. Bacterioids, represented in the blue, are negatively associated with depression symptoms, meaning that the lower levels in the gut are linked to higher symptoms of depression. Faecalibacterium, in the orange, shows a positive association, or higher levels of this taxa in the gut are linked to fewer symptoms of depression. The third bar in green represents other taxa with a weaker positive association to depression.
Pritchett et al. Investigated the potential link between antibiotic use and risk of developing mental health disorders such as anxiety and depression within a primary care health setting. This retrospective cohort study included pediatric subject sample size of 5,244 between the ages of 8 and 10. The researchers focused on patients who were prescribed antibiotics, trying to assess whether exposure to antibiotics increased the likelihood of future mental health diagnosis within a 5-year period. Multivariate Cox proportional hazard model, a statistical model used, was to evaluate the risk of depression diagnosis while accounting for potential confounding factors like age, race, sex, and insurance. The findings revealed a potential association, particularly highlighting that individuals treated with broad-spectrum antibiotics were at a greater risk of developing mental health conditions compared to those who did not receive antibiotics or were treated with narrower-spectrum drugs.
The study in 2022 in China by Tian et al. Looked at 45 patients with major depressive disorder, and they determined they had major depressive disorder through standardized instruments. They had a double-blind randomized control trial that looked at 20 patients that consumed 1,010 CFUs of a specific type of bifidobacterium, strain CCFM1025, and 25 people received placebo for 4 weeks. They measured depressive symptoms, gastrointestinal symptoms, serum serotonin turnover, fecal microbiome, and tryptophan metabolomics. Tryptophan is related to serotonin production by our gut, and if there's higher serotonin turnover, then serotonin is also getting metabolized by our gut microbiota. So what they found was that depression scores were significantly reduced in groups where Bifidobacterium breve was administered. They also found that Bifidobacterium breve helped significantly in increasing microbiotic diversity and significantly reduced serotonin turnover, as well as upregulating some of the tryptophan metabolites that they studied.
These are our results from the study review. All the studies did examine the link between antibiotics, gut microbiome, and depression. 2 of the studies focused on gut microbiome composition and its links to depressive symptoms. Some key findings in these studies was antibiotics disrupted gut microbiome diversity. Increased antibiotic-resistant genes were strongly associated with a higher risk of depression. Specific gut microbial taxa was linked to depressive symptoms. Higher microbiome diversity was protective against depression. And a recurrent theme we noticed throughout the studies were disruptions in gut microbiota via antibiotics or altered microbiome composition are linked to adverse mental health outcomes, particularly in depression.
One common theme we found throughout all of our studies was that microbiome disruption was linked to depression, and that could have been through antibiotic use or also having decreased microbial diversity attributing to depression. And they looked at specific taxa in some of the articles we looked at, and there were a few that were linked to depression. Of note, I would say Clostridium and Egerthella were linked to depression, and because of their incidence, they were also linked to disruption because they would overtake the gut microbiota, and there were not as many species when these were found. And all of our studies looked at fecal matter sampling to assess gut microbiome composition, and on all of our studies were on mixed genders, and age groups were the same except for one group where they used a pediatric group. Some contrasting factors between our studies were that the depression scales were different among the studies, so it's difficult to determine if there was as big of an effect between different studies. Different antibiotics were used in some of these studies, and they found different microbial taxa were affected by these different antibiotics, so it's hard to tell if one specific has a long-term effect. And so some of our studies looked at short-term effects of microbiome disruption, and some looked at long-term effects, so it's hard to prove causation in some of these longer-term studies.
There were some limitations to 6 studies that were reviewed. The retrospective study designs were limited in establishing direct cause and effect relationship due to its retrospective data. The depression measures in Tian et al. Presented potential bias risk by relying on self-reported questionnaires, which can be subjective and influenced by external factors. Microbiome analysis focused on specific microbial species due to budgetary constraints. This potentially overlooked a more nuanced relationship between specific microbes and depression. In Pritchard et al., population differences focused on a homogeneous pediatric population when compared to the other 5 studies, which focused on a heterogeneous adult population. This added a layer of complexity and limited generalizability when comparing findings.
In conclusion, these studies have shown antibiotic use and imbalances in gut microbiome are strongly linked to depression. The reviewed studies also suggest that restoring microbial balance may help reduce depressive symptoms, highlighting the gut-brain axis as a potential target for future mental health treatments. Applications: designing dietary recommendations as interventions that support gut health to potentially reduce the risk of depression, and promoting awareness about mental health implications of antibiotic use, encouraging practices that preserve gut health. Future research in this area: investigating the biological mechanisms through which gut microbes influence mood and behavior, research into the gut-brain axis to explore other psychiatric disorders linked to microbiome alterations, and developing standardized methods for assessing mental health outcomes related to gut health.